Welcome to the deep dive. Today we're diving into a world that you might not expect to be so fascinating.
Yeah,
The surface of a nematode.
it's true. We often think of lipids as those fats, tucked away inside organisms, but this 2025 research from the Journal of the American Chemical Society, it reveals that they're doing some truly remarkable things on the outside of these tiny creatures.
Okay, so for anyone who needs a quick reminder, nematodes, they're roundworms and they're incredibly diverse and abundant.
Mm-hmm.
We're talking about species like C. elegans, which is really a workhorse in labs because we can use it to understand fundamental biology.
Right.
What makes this research so groundbreaking is that it focuses on the nematode cuticle.
Exactly. The cuticle is this tough outer layer, and it's
So
protective.
it's like armor.
It is like armor. It shields the nematode from all sorts of things, like drying out pathogens and even other predatory nematodes.
Oh my gosh.
It's really their first line of defense against the world.
So it's like a microscopic suit of armor.
Yes.
But this research goes deeper using an advanced imaging technique called 3D orbisims.
Right.
What's so special about that?
With 3D orbisims, it allows scientists to map the chemical composition of the nematode's surface
Okay.
in incredible detail and all without having to cut them open or damage the cuticle.
Wow.
Think of it like having molecular X-ray vision.
That's amazing. And what did they discover with this X-ray vision? Did they find anything unexpected?
They found that lipids actually dominate the nematode surface.
Oh, wow.
And that was a surprise because we typically think of lipids as being stored inside the body.
Right,
They uncovered a diverse array of lipids like fatty acids,
right.
triglycerides, and even sterols, which are similar to the cholesterol that we have in our bodies.
So it's not just a simple greasy coating, it's a complex mixture of different lipids.
Yeah,
What's really intriguing is that the researchers found this lipid composition changes as the nematode develops.
yes, the surface of a larval nematode is very different from that of an adult.
Mm.
For example, both C. elegans and P. pacificus, they show a marked increase in longer-chain fatty acids and triglycerides as they mature.
That makes me wonder why the change. Is there an evolutionary advantage to having different lipids at different life stages?
That's a great question, and the researchers believe these changes reflect adaptations to their environment and lifestyle.
Okay.
Think about it, a larva might need a certain set of lipids to help it grow and develop,
Uh-huh.
while an adult might need different lipids for reproduction or protection.
That makes sense. It's like their surface adapts to the challenges they face at each stage of life.
Right.
But is this lipid profile just about development, or is there something more to it?
Well, remember how we talked about nematodes being incredibly diverse?
Yeah.
It turns out their surface lipids reflect that diversity as well.
Mm-hmm.
The researchers compared the surface profiles of C. elegans, which typically hangs out in rotting fruit,
Okay.
to P. pacificus, which is often found associated with beetles.
Okay, I'm on the edge of my seat here.
And what they found was fascinating.
What did they find?
They discovered that these two species have distinct lipid fingerprints on their surfaces.
Wow.
This suggests that the lipids are not just a random assortment,
Yeah.
but are specifically tailored to the nematode's ecological niche.
Oh, wow.
So C. elegans might need certain lipids to thrive in the sugary, acidic environment of rotting fruit,
Okay.
while P. pacificus might need different lipids to interact with the waxy exoskeleton of a beetle, for example.
So the lipids are like a chemical signature telling us something about where the nematode lives and how it interacts with its environment. It's incredible how much information is packed into those tiny molecules.
Yeah.
But how are these lipid profiles created? Is there some sort of control center for this process?
There is. The researchers uncovered a key player called the DAF22 gene.
Okay.
This gene is involved in a crucial pathway called the peroxisomal ioxidation pathway,
Okay.
which is like a cellular recycling plant for lipids.
Oh.
It breaks down fatty acids and also produces important signaling molecules
Okay.
that can influence various processes within the nematode.
So DAF22 is like the master conductor of the lipid orchestra. What happens if you disrupt this conductor?
Well, the researchers did just that.
Oh.
They mutated the DAF22 gene in both C. elegans and P. pacificus to see what would happen.
Okay.
And the results were quite dramatic.
I bet. Messing with a gene that controls something as fundamental as lipid metabolism is bound to have some consequences.
Yeah, but with some interesting twists.
What did they observe?
In C. elegans, mutating DAF22 led to a significant drop in several key lipids on the surface, including triglycerides and sterols.
Okay.
This suggests that the peroxisomal E-oxidation pathway plays a critical role in maintaining the normal lipid balance in this species.
So disrupting DAF22 really threw things out of whack for C. elegans.
It did.
What about P. pacificus? Did it have a similar meltdown?
Surprisingly, not quite.
Okay.
While the P. pacificus mutants did show some changes,
Uh-huh.
they didn't experience the same dramatic decrease in overall lipid content. This leads researchers to think that P. pacificus might have some sort of backup mechanism
Oh,
to keep its lipid levels stable even when the peroxisomal aboxidation pathway is disrupted.
it's so interesting. It's like P. pacificus has a plan B for its lipids, while C. elegans is more reliant on that one pathway.
Right.
I wonder if that has something to do with their different lifestyles and the environments they've adapted to.
It's certainly a possibility, and
Yeah.
it highlights the incredible diversity and resilience of these tiny creatures.
Yeah.
Even at the molecular level, we see different species evolving unique strategies to survive and thrive.
It's a reminder that even seemingly simple organisms like nematodes can have surprisingly complex biology.
Yes.
But why is all of this so important? Why should we care about the lipids on the surface of a worm?
Well, remember how we said the cuticle is the nematode's first line of defense?
Yeah.
The researchers believe that these surface lipids play a crucial role in protecting nematodes from predators.
Okay, now we're talking about a real-world application. So these lipids aren't just there for show, they're part of a sophisticated defense system.
Right.
What kind of experiments did they do to test this?
They set up what you might call a microscopic gladiatorial arena.
Uh-huh.
They pitted the predatory nematode P. pacificus against C. elegans
Okay.
to see if changes in C. elegans' surface lipids would affect its vulnerability to being eaten.
So it's a microscopic battle for survival. What happened? Did the lipids make a difference?
There absolutely did.
Wow.
The C. elegans mutants with the disrupted lipid profiles were much more likely to become a meal for P. pacificus.
It's like their messed up lipids made them easier targets.
Right.
So the normal surface lipids in C. elegans might actually be helping them avoid detection by predators or even make them harder to attack.
Precisely. It's a fascinating example of how even tiny changes at the molecular level can have significant consequences for an organism's survival.
Wow.
And this research raises some intriguing questions about the role of surface lipids in other organisms, even us.
That's a great point. It makes you wonder if the lipids on our own surfaces play a role in how we interact with the world around us.
Yeah.
From attracting or repelling microbes to influencing our susceptibility to diseases.
Right.
I can't wait to dive deeper into these possibilities in the next part of our deep dive.
It really makes you wonder if we've been overlooking the importance of surfaces for far too long.
Yeah.
They're not just static barriers, but dynamic landscapes that play a critical role in how organisms interact with their environment.
Absolutely. We've been so focused on what's going on inside cells and organisms that we've neglected the fascinating world happening right on the outside.
Right.
Speaking of those predator-prey experiments, did the researchers uncover how those altered lipids in the C. elegans mutants made them more vulnerable?
That's a question they're still exploring,
Okay.
but they do have some intriguing hypotheses.
Oh,
One possibility is that the missing or altered lipids weaken the structural integrity of the cuticle,
okay.
making them more susceptible to physical attack.
So it's like their armor lost its strength and became easier for the predator to pierce. Makes sense.
Exactly. Another fascinating possibility is that the changes in surface lipids altered the chemical signals C. elegans,
Okay.
making them more detectable to the predator.
So it's like their camouflage failed and they were suddenly standing out like a beacon to the predator.
Indeed. It's also possible that the changes in surface lipids disrupted other important processes on the nematode surface,
Okay.
like their ability to sense their environment or defend against pathogens.
Uh-huh.
These disruptions could indirectly make them more vulnerable to predation as well.
Wow, so many possibilities. It seems like these surface lipids are involved in a whole network of interactions we're just beginning to unravel.
Absolutely. It's a testament to the power of cutting-edge technology like 3D orbisms, allowing us to see things at a level of detail that was previously unimaginable.
And as we discussed earlier, this research has implications that go far beyond the world of nematodes.
Right.
It makes you wonder if similar processes are happening on the surfaces of other organisms, even as humans.
Precisely. Think about our skin, our gut lining, our lungs. All these surfaces are in constant contact with the environment, teeming with microbes and molecules.
So could the lipids on our surfaces be playing a role in how we interact with the world around us?
Yes.
Could they be shaping our microbiome or influencing our susceptibility to disease?
It's certainly a possibility worth exploring.
Okay.
We already know the composition of our skin microbiome can influence our health in various ways.
Mm-hmm.
It's conceivable that the lipids on our skin surface play a role in shaping that microbiome, either by attracting or repelling certain types of bacteria.
That's incredible. So like nematodes, We could have unique lipid fingerprints on our skin that influence which microbes decide to call it home.
Exactly, and the implications are far-reaching.
Yeah?
Could altering the lipid composition of our skin help us fight off infections,
Uh-huh.
or even treat skin conditions like eczema?
It's mind-blowing to think that something as seemingly mundane as the lipids on our skin could have such profound effects on our health.
And that's just the tip of the iceberg. The study of surface lipids is still a relatively new field,
Okay.
with countless questions waiting to be answered.
Well, one thing's for sure. We can't ignore the surface anymore. It's
Right.
a dynamic and essential part of biology, and it holds the key to understanding a whole range of processes from development to disease.
Speaking of disease, one area where the study of surface lipids is showing particular promise is in the fight against parasitic infections.
Now that you mention it, I do recall reading something about how parasitic nematodes use their surface lipids to evade the host's immune system.
That's right. Parasitic nematodes, like the ones that cause diseases like hookworm and elephantiasis,
Right.
are masters of disguise.
Oh, wow.
They've evolved sophisticated strategies to coat themselves in lipids that mimic those of their host,
Oh,
making them invisible to the immune system.
they're like microscopic wolves in sheep's clothing, blending in with their surroundings to avoid detection.
A very apt analogy. This has enormous implications for developing new treatments for these devastating diseases.
Yeah.
If we can figure out how to disrupt the lipid camouflage of these parasites,
Uh-huh.
we could make them vulnerable to the host's immune system.
It's like taking away their invisibility clothes and making them easy targets for our body's defenses. That's a brilliant strategy.
Exactly. And there's already exciting research underway in this area.
Oh, wow.
Scientists are developing drugs that target specific enzymes involved in lipid synthesis in parasitic nematodes.
Okay.
The goal is to disrupt their ability to produce the lipids they need for camouflage and survival.
So it's like a microscopic arms race with scientists constantly trying to outsmart these parasites and their ever-evolving lipid armor.
It certainly is, and it highlights the importance of understanding the fundamental biology of these parasites, including their surface chemistries.
Speaking of fundamental biology, I'm curious to know more about that peroxisomal eoxidation pathway we touched on earlier.
Okay.
You mentioned it plays a critical role in shaping the surface lipid profile, but what exactly is it and why is it so important?
The peroxisomal eoxidation pathway is a metabolic process that takes place inside tiny cellular compartments called peroxisomes.
Okay.
Think of it as a highly specialized recycling plant for fatty acids.
Okay, so we have these tiny recycling plants inside our cells dedicated to breaking down fats.
Right.
What exactly happens in these recycling plants?
Well, long-chain fatty acids, the building blocks of many lipids, are transported into the peroxisome and broken down into smaller molecules.
Okay.
These smaller molecules can then be used for a variety of purposes.
Uh-huh.
Some are used to produce energy for the cell, while others are transformed into important signaling molecules
Okay.
that regulate various processes like development, metabolism, and immunity.
So it's not just about breaking down fats, it's about creating valuable building blocks for other essential molecules.
Precisely. And the DAF22 gene we discussed earlier encodes an enzyme that plays a key role in this breakdown process.
Okay.
When you mutate DAF22, you're essentially disrupting the entire recycling plant,
Right.
which explains why it had such a dramatic effect on the surface lipid profiles, particularly in C. elegans.
It all makes sense now. It's like a delicate balancing act with all these different pathways working in harmony to keep the organism functioning properly.
And when that balance is disrupted, either by genetic mutations or environmental factors,
Right.
it can lead to a whole range of problems, including disease.
It's amazing to think that these tiny microscopic processes can have such enormous macroscopic consequences.
It really speaks to the complexity and elegance of biology.
Absolutely. The more we learn about these intricate processes, the more we appreciate the wonders of the natural world.
And the more possibilities we uncover for developing new treatments and therapies to improve human health.
Speaking of possibilities, I'm eager to learn more about those drugs that target lipid synthesis in parasitic nematodes. Are they showing any promise in combating these infections? Welcome back to the Deep Dive. We're wrapping up our exploration of nematode surface lipids, and I have to say it's been a real eye-opener.
Yeah, it's a fascinating area.
Who knew there was so much going on at the microscopic level?
It's truly remarkable, isn't it? We've gone from the intricate details of lipid molecules to the grand sweep of evolution, uncovering surprising insights along the way.
Absolutely. And speaking of surprises, I'm still reeling from the idea that surface lipids could be playing a role in shaping the microbiome,
Right.
not just in nematodes, but potentially in humans as well.
It's a fascinating concept and one that researchers are actively investigating.
Yeah,
Imagine if we could manipulate the lipids on our skin to cultivate a healthier microbiome.
yeah.
It could lead to entirely new approaches for preventing and treating skin infections,
Wow.
boosting our immune system, and even addressing conditions like acne or eczema.
That's incredible. It's like we're discovering a whole new dimension of human health right there on the surface.
Precisely. And that's just one example.
Yeah.
The study of surface lipids is opening up new avenues of research in diverse fields from agriculture and materials science.
Speaking of diverse fields, I'm particularly intrigued by the evolutionary insights we can glean from studying surface lipids.
Right,
We touched on it earlier, but I'd love to delve a bit deeper.
of course. As we discussed, the specific composition of surface lipids can vary dramatically between species, reflecting adaptations to different environments and lifestyles.
Right.
For instance, the lipid profiles of a nematode that lives in the soil would be vastly different from those of a nematode that lives inside a beetle.
It's like each species has its own unique lipid fingerprint,
Yes.
a chemical signature that tells a story about its evolutionary history.
Exactly. It's like piecing together a puzzle using lipid molecules as the clues.
Yeah.
And the more pieces we find, the clearer the picture becomes.
Uh-huh.
For example, researchers have used lipid analysis to study the evolution of parasitism in nematodes. They've discovered that parasitic species often have unique lipids on their surface that help them evade the host's immune system.
It's a fascinating example of how evolution can drive the development of sophisticated molecular strategies for survival,
Right.
and it highlights the importance of understanding these strategies if we want to develop effective treatments for parasitic infections.
Absolutely. But the evolutionary implications of surface lipids extend far beyond parasitism.
Okay.
They can provide insights into the origins of multicellularity,
Wow.
the development of specialized tissues, and even the evolution of social behavior.
It's incredible to think that these tiny molecules can tell such grand evolutionary stories.
It really speaks to the interconnectedness of life on Earth and the power of molecular biology to illuminate the hidden connections between the past, present, and future of life itself.
Well, we've covered a lot of ground in this deep dive, from the microscopic details of lipid molecules to the vast sweep of evolution.
We have.
As we wrap up, what are your key takeaways from this exploration?
For me, the most striking takeaway is the realization that surface lipids are not just passive components of an organism's biology. They are dynamic, essential, and constantly evolving to meet the challenges of their environment.
They are a critical interface between the organism and its environment, playing a vital role in everything from development to defense to communication.
And as our understanding of surface lipids continues to grow, we are bound to uncover even more fascinating and unexpected insights with profound implications for human health, agriculture, and even our understanding of the evolution of life itself.
This has been an incredible journey. Thanks for joining us on this deep dive into the world of nematode surface lipids. We hope you've learned something new and that you'll continue to explore the wonders of the often overlooked surface.
Until next time, keep diving deep.